former PhD student at Abate Bashaw. He spent nearly 4 years in the lab doing a PhD with us. He came originally from Ethiopia, and there he did his undergraduate degree at Tafasa University, and then moved to the University of Gondar to do a master's degree. He also taught as a lecturer in the University of Gondar, Ethiopia, and he was there as a teaching microbiology and immunology before he came to do his PhD with us. Abate has been a great asset around the lab. He's been one of the nicest students I've ever had to work with. He's considerate of others. He gets on with his work, and he comes and asks nicely for things rather than coming and demanding them, which some students have been known to do. So he had, I think, a very successful PhD thesis, as evidenced by the fact that he was offered several jobs after he finished. And he's happened, fortunately for this morning's performance, to take one in Australia rather than overseas, where he might have been. And he's going to tell us about the work that he actually did during his PhD thesis, answering an important question relating to the way that the virus, papillomavirus, suppresses the local immune system to enable its long-term survival. Over to you, Abate.
Okay, thank you so much, Ian, for your nice introduction. Really grateful. And good morning, everyone. And thank you for the opportunity. So today, I'm going to present probably a brief summary of my PhD studies. As Ian mentioned, my PhD concentrates on understanding the immune suppressive mechanisms of HPV-associated neoplastic disease, especially initiation of primary immune responses. The title for my today's presentation is HPV16 E7-mediated epithelial hyperplasia impairs cell-mediated immune responses through inducing peripheral regulatory T cells. Just to introduce a little bit about HPV infection and associated diseases. HPVs are responsible for various types of skin and mucous membrane lesion. Majority of HPV infection are cleared by the immune system within 1 or 2 years, but in certain individuals, which is about 10 to 15% of infected women, the virus can invade the host immune system and establish persistent infection, which results in epithelial dysplasia, probably in about 1% of infected individual leads cervical cancers, the invasive cervical cancers. So far, over 200 HPV types have been identified, of which at least 10 of them are high-risk oncogenic, responsible for HPV-associated carcinogenesis. However, type 16 and 18 are the frequent cause of precancerous and cancerous HPV-associated lesion. For instance, about 50% of cervical cancers have been linked with HPV type 16, while 15% of cervical cancer linked to type 18. Despite the tremendous success in the prevention of cervical cancer through the prophylactic vaccination and the cervical cancer screening programs, still the disease is remaining a significant public health problem, especially in middle income and low and middle income countries, due to the limited vaccine intake and lack of implementing the cervical cancer screening program. This, the bottom diagram or the bottom graph indicated the number of cases per 100,000 women across different age groups and different economic countries like low, middle, and high income countries. HPV normally infects the basal keratinocytes, and during persistent infection, overexpression of the high-risk HPV type E6 and E7 oncoproteins within the infected epithelial cells are the drivers of cell transformation and neoplastic progression. These E6 and E7 oncoproteins are able to transform the infected epithelial cells through inactivating the cell cycle regulatory proteins like P15-3 and erythroblastoma protein. E6 and E7 proteins are constitutively expressed in epithelial cells during precancerous and cancerous lesion, making them the ideal target for developing therapeutic vaccines. However, despite a huge efforts, therapeutic vaccine targeting these oncoproteins are not yet successful in the clinic, and the immune suppressive local microenvironments are thought to be responsible for vaccine failure. And therefore, to understand the immune suppressive mechanisms and develop potential therapeutic HPV vaccine, Professor in Fraser Lab at the University of Queensland Diamantina Institute has established a mouse model expressing the type 16 E7 oncoprotein in skin keratinocytes under the control of keratin 14 promoter. And this transgenic mouse model expression of E7 protein alone leads epithelial hyperplasia, which mimics the precancerous lesions of HPV positive human tissue, which is characterized by hyperproliferation of keratinocytes, which results in thickening of skin epidermis, which induces infiltration of different cells and induce chronic inflammation, which are associated in local immune suppression. Unlike other self-antigens expressed by mouse keratinocytes, like ovalbumin, E7 expressing skin are tolerated when transplanted into wild-type immunocompetent hosts, indicating the local immune suppressive properties of this E7 expressing skin. And different chronic inflammatory cytokines like IL-17 and interferon gamma and cellular infiltrators such as invariant NKT cells have been contributed to the immune suppressive natures of this hyperplastic skin. And Langerhans cells, which are the predominant antigen presenting cells in the epidermis, exhibited atypical phenotype. They express significantly low levels of antigen presenting molecule like MHC class 2, and they have less dendritic morphology, which indicates the alteration of local antigen presenting cells. This alteration of antigen presenting cells is correlated with clinical studies where the intraepithelial Langerhans cells have been reported to be altered by HPV-induced epithelial hyperplasia. As we know, diseases are very critical for orchestrating innate and adaptive immune responses. In a steady state, skin disease include Langerhans cells in the epidermis, and CD11B positive and CD103 positive dermal disease in the epidermis. These are highly specialized antigen presenting cells due to their capacity to migrate to the draining lymph node and prime naive T cells. They are required to capture antigens from the peripheral tissue and transport the antigen and present the antigen to T cells in the draining lymph nodes so that the T cells undergo clonal expansion and differentiation into a particular effector T cell subtypes. And the nature of antigens and the nature of antigens and activation status of antigen-bearing dendritic cells, as well as the inflammatory environment, and the presence of other responding T cells in the draining lymph nodes determines the fate of antigen prime T cells. For instance, antigen presentation by immature disease in a steady state favors differentiation of T cells into regulatory and energetic T cells, which are very important to maintain peripheral immune homeostasis and prevent autoimmune disease. However, under inflammatory conditions, dendritic cells, antigen-bearing dendritic cells undergo functional maturation. They upregulate the antigen-presenting molecule, including MHC class 2 and the co-stimulatory molecule CD8086, and produce pro-inflammatory cytokines like IL-12, IL-1 beta, and IL-6. Therefore, antigen presentation by such mature DC induces effector T cell differentiation, such as Th1 and Th2 or Th17. These types of effector T cell responses primarily depends on the nature of antigens. So as dendritic cells are very crucial to initiate primary immune response, and modulation of disease by tumor microenvironment often impairs the anti-tumor immune responses. Regulation of dendritic cells by HPV-associated neoplastic environment might be one of the mechanisms that suppresses the immune response. Therefore, the overall hypothesis of my project was E7-driven epithelial hyperplasia in the transgenic mouse model induces peripheral T cell tolerance through modulating local antigen present in cell development and their antigen presentation function. And the main aims of my project were to examine the impact of E7-driven epithelial hyperplasia on DC antigen presentation and priming of T cells, and to investigate the mechanism by which E7-driven hyperplasia modulates disease and T cells, especially helper T cell responses. So first, we examined the phenotypes of skin disease. These are the typical getting strategies we used to identify skin disease subsets. The hematopoietic marker CD45 positive cells that co-express high levels of CD11C and MHC class 2 were considered to be skin dendritic cells, which further divided into dermal and epidermal disease subsets. As expected, the wild-type epidermis comprises only Langerhans cells, while the E7 expressing hyperplastic skin additionally contains the dermal DC subsets like CD11P positive disease and CD103 positive disease. And the total number of dendritic cells in E7 expressing skin were significantly higher compared to control hosts. Most importantly, the antigen presenting molecule class 2 were significantly lower from the surface of Langerhans cells isolated from E7 expressing skin, while the co-stimulatory molecules like CD80 and 86 were upregulated. So this infiltration of epidermis with dermal DC subpopulation and increased number of total dendritic cells and upregulation of the co-stimulatory molecules indicates the impact of hyperplasia associated chronic inflammation. But down regulation of the class 2 expression indicates the regulation or suppression of local antigen presenting cells. Therefore, to test whether this surface expression, lower surface expression of MHC class 2 molecule is associated with the biosynthesis of MHC class 2, we measure the intracellular MHC molecules. Unlike the surface levels, this E7 expressing skin disease have accumulated in MHC class 2 molecule in the intracellular compartment, and none of the genes encoding MHC class 2 related molecules are down-regulated by class 2, E7 expressing hyperplastic environment. This suggested that the biosynthesis of class 2 molecule is not down-regulated. Rather, the chronic environment might cause instability of MHC class 2 surface expression. Sorry. So next, to determine whether these phenotypic differences affect the function of skin dendritic cells, we examine the antigen uptake and activation properties of disease after immunization. So we immunize the mice with the fluorescent dye F488 conjugated ovalbumin and the presence of immunostimulatory adjuvant chelae. And 24 hours later, the skin and skin draining lymph node disease were examined for the antigen uptake and upregulation of activation molecules. So the results show that dendritic cells derived from E7 expressing skin have limited capacity to uptake antigen. And most importantly, those diseases that have taken up the antigen failed to upregulate the antigen presenting molecule MHC class 2 and co-stimulatory molecules like CD86, indicating the impaired functionality of E7 expressing skin disease. Therefore, to test if this phenotypic or the reduced antigen uptake and activation properties of E7 expressing skin limits their capacity to prime T cells in vivo, we transferred overreactive CD4 T cells, which is labeled with the proliferation dice FSE. And after immunization with OVA and the adjuvant chelae, that proliferation and activation of T cells were determined. Surprisingly, the T cells transferred into E7 expressing animals undergo proliferation almost similar to wild type hosts and express significantly high levels of T cell activation marker like CD44 and CD69. But these proliferating T cells failed to produce effector cytokines like the type 1 Th1 cytokine interferon gamma upon in vitro restimulation. And only very few proliferating CD4 T cells produce interferon gamma, but this chelae and OVA immunization didn't induce production of other helper T cell cytokines like IL-10, IL-17, and IL-4. So to examine whether these proliferating T cells would acquire regulatory phenotypes after adoptive transfer and subsequent to immunization with 1 week interval, the transfer T cells were examined for expression of anergic and regulatory markers. And significantly, the results show that significantly higher numbers of transferred T cells into the E7 expressing animal acquire FOXP3 positive Tregs. And those FOXP3 negative T cells co-express high levels of T cell energetic marker, CD73 and FR4. T cell energy is a state of immune responsiveness in which primed T cells undergo proliferation and incomplete activation, but upon restimulation, they are unable to produce effector cytokines and growth factors. From the results that I have presented so far, we can conclude that in vivo antigen priming by E7 expressing antigen present cells after intradermal immunization efficiently stimulates CD4 T cell proliferation, but the proliferating cells do not secrete effector cytokines. Instead, the proliferating cells acquire energy and regulatory T cell phenotypes. And the next question was what drives regulatory or energetic T cell differentiation in this E7 expressing hyperplastic hosts. So first, to exclude the hyperplasia independent effect of E7 expressing, we have used E7 expressing non-hyperplastic mice. In these mice, keratinocytes express the mutant form of RB, which is selectively defective for binding to E7 protein, so that it has normal cell cycle regulatory functions and normal epithelial proliferation. In normal cells, binding of RB to the host transcription factor A2F is required to control cell proliferation, but during HPV infection from high risk types, E7 binding to the RB protein disrupts the binding of RB to the transcription factor A2F, which leads to uncontrolled cell proliferation and which results in epithelial dysplasia. In this E7 expressing non-hyperplastic mice, CD4 T cell is primed by this non-hyperplastic E7 expressing skin disease, produce efficiently interferon gamma, comparable to the control mice, and no phenotypic differences were observed between this E7 expressing hyperplastic skin disease and the control mice, which indicated that it is the hyperplasia or E7 RB interaction resulting in immune suppression, but not the E7 expressing alone. Although it has not been elucidated the mechanisms of immune suppression, NKT cells through production of interferon gamma has been shown to contribute to the immune suppressive properties of E7 expressing skin. Of course, interferon gamma, recent evidence supports the regulatory effects of interferon gamma, and in chronic tumor microenvironment, interferon gamma can induce IDO expression, IDO and PDL1 expression in antigen presenting cells, including the tumor cells, so that it induces Treg expansion and leads to exhaustion of tumor infiltrating T cells. Therefore, to determine whether the interferon gamma or NKT cells contribute to the immune suppression in E7 expressing animals, we used interferon gamma and NKT cell deficient mice. In these mice, surprisingly, we found that neither NKT cells nor interferon gamma deficiency restores T cell immune response. But interferon gamma and NKT cell deficiency also significantly compromises Th1 response in control non-E7 transgenic mice, indicating the significant roles of interferon gamma in generating adaptive immune responses. Of course, interferon gamma is very important to generate adaptive immune response. Interferon gamma produced by innate immune cells, including NKT cells, are very critical to generate adaptive immune response in normal immune conditions. And sorry. The E7 expressed, this E7 expressing K4-E7 mice not only express the E7 protein in the skin keratinocyte, but also in the thymus epithelial cells, resulting in thymus hypertrophy. We know that thymus epithelial cells are very important to program T cell development. Therefore, these T cells developed within the hypertrophic thymus may become very prone to acquire immune suppressive properties upon immunization. Therefore, to exclude this, the potential effects of this endogenous thymus-derived regulatory lymphocytes on peripheral T cell tolerance, we have used the E7 expressing RAG knockout mice, which lacks both B and T lymphocytes. And after adoptive transfer of purified CD4 T cells, 3 weeks later, we found that the majority of transferred T cells acquired the FOXP3 positive Tregs. But this hyperplasia-induced immune suppression do not control the cytokine production by the transferred T cells, which indicated the role of lymphopenia-induced pathogenic T cell generation. We know that hyperplasia lymphopenia can revert even allergic T cells to effector T cells that produce effector cytokines, even in the presence of tumor. So to specifically address the question whether E7 expressing skin modulates directly the T cell priming in the draining lymph nodes, we transplanted either 2 E7 or wild type skin into wild type hosts. And after adoptive transfer and intragraft or ear immunization with OVA and Cule, the proliferation and CD4 T cell, FOXP3 expression was measured. And the results showed that a significant number of transferred T cells acquire FOXP3 positive Tregs in hosts which transplanted with E7 expressing skin. This indicated that this demonstrated that the E7 expressing hyperplastic skin directly modulates immune response in the peripheral lymphoid organ. So previously, it has been shown that from the gene expression data, IL-10 expression was significantly higher in E7 expressing skin compared to wild type skin, which indicated the role of IL-10 in E7 expressing animals. We know that IL-10 is very important regulatory cytokine, which can directly control effector response or indirectly through regulating antigen presenting cells. So to determine whether IL-10 signaling in E7 expressing host induces immune suppression, we have used IL-10 deficient E7 transgenic mice. But surprisingly, the results show that CD4 T cells transferred into IL-10 knockout mice acquires FOXP3 positive T cells almost similar to that of IL-10 reflect E7 transgenic hosts. And the cytokine production of the transferred T cells were significantly impaired in these IL-10 deficient mice. Both IL-10 sufficient and IL-10 deficient E7 transgenic mice also contain significantly higher numbers of endogenous FOXP3 regulatory T cells that express CD25, CD44, CD73, and CTLA4 in the skin. So next to determine whether the antigen presenting cells are programmed to induce T cell tolerance, we immunize the mice with fluorescent labeled antigen, and 48 hours later, skin dendritic cells and draining lymph node antigen positive dendritic cells were isolated by FACS and co-culture with purified CD4 T cells for 5 days and analyzed for the phenotype and effector cytokine production. Strikingly, the result indicated that unlike the in vivo priming results, antigen presentation by E7 expressing skin-derived disease ex vivo efficiently induces interferon gamma production by CD4 T cells, even though there was slightly increased FOXP3 expression and E7 expressing a skin DC co-culture system. So the possible explanation for the discrepancy between the in vivo and ex vivo results might be the soluble immune mediators may be required to control the functionality of skin-derived disease in the draining lymph nodes, or the dendritic cells may regain their immunostimulatory function once they are removed from E7 expressing immune suppressive environment, or the soluble immune regulatory cytokines or factors might be required to directly induce immune tolerance in the draining lymph nodes. So finally, to examine the functional relevance of peripherally expanding regulatory CD4 T cells, we depleted the CD4 T cells and examined the proliferation and effector cytokine production of both CD8 and CD4 responses. In this experiment, prior to depletion of CD25 positive Tregs, we immunized the mice with OVA and QLE intradermally to build up peripheral regulatory T cells, and then 2 days before and after OT2 cell transfer, the CD4, OT2 or OT1 T cell transfer, we treated the mice with anti-CD25 antibody, and after second immunization, the proliferation and activation of CD8 and CD4 T cells were examined. The result indicated that this CD25 positive Treg depletion restores significantly the proliferation and effector cytokine production by CD8 T cells. But this regulatory T cell depletion didn't restore the Th1 response, indicating the presence of multiple immune suppressive pathways. Therefore, from this study, we can conclude that this type 16 E7-driven epithelial hyperplasia down-regulates antigen uptake and activation capacity of skin dendritic cells, and antigen presentation through intradermal immunization skewed CD4 T cell response toward this regulatory and T cell energy. These Tregs are responsible to suppress CD8 T cell response, but depletion of Tregs does not suffice to restore Th1 responses. However, as dendritic cells are functionally competent ex vivo, it is not yet clear whether the modulation of antigen-presenting cells contribute to drive regulatory T cells in vivo. So finally, I would like to acknowledge my supervisor, all my supervisors, Professor Impairs, Dr. Jenny Chandra, and Professor Graham Laggett for their invaluable support and guidance. They have been extremely supportive mentor. I'm so humble and grateful for that. I also thank my thesis committee, Associate Professor Ramon Steto, Professor Matthew Sweet, Associate Professor Christine Rafford, for their time and support throughout my candidature. I would like to also thank the past and present Fraser Lab groups for their help and kindness throughout my stay in the lab. And I would like to also thank the core facilities, especially the biological research facility and flow cytometry facility, for their technical support, and the UQ International Scholarship for the financial support. Thank you for listening, and I'm happy to take some questions.